You're listening to an Airwave Media Podcast.
Welcome to Unbiased Science,
where we bring scientific method to the madness.
We're your hosts, Dr. Steier
and Dr. Sarah Scheinman.
And this week we are going to talk about mRNA, because those four letters are at the tip of everyone's tongues these days. And we're joined by a very special guest, Dr. Nancy Haigwood. I'm going to introduce her to you in just a moment, and her incredible background, expertise, and experience that is super relevant to this topic. But before we do, if you recall, we are now kicking off every week with just a really brief recap of what's going on in public health science news. So I wanted to talk about two things. The first is that last week, the Wall Street Journal broke the news that HHS is expected to release a report, that big autism report that we're all waiting for with bated breath, and that they might point to Tylenol exposure in utero as a potential, cause of autism. And I'm choosing my words really carefully here because, as we've discussed, of course, autism is multifactorial. It is already very well established that it's primarily driven by genetic factors. However, we're all now expecting this big report to point to Tylenol given or Tylenol exposure during pregnancy. Now, I just want to call attention to the fact that there is a lot of research done on this topic. There's this great study out of Sweden that did an incredible job controlling for the genetics by including siblings in their research study that found no association between Tylenol exposure and pregnancy and autism or ADHD and a number of other outcomes. And so the body of evidence that we've evaluated thus far at Unbiased Science does not point to this, but it's tricky and potentially harmful because we know that things like fever during pregnancy carry their own risks, right? So if you have a high fever while pregnant, that can cause neural tube defects, spina bifida, and other things. So we really have to be careful when messaging around this. So just wanted to flag this for you all. We did do a Substack, and we're going to keep investigating, and we'll report on more as this report gets released. The other big thing that happened is that HHS released their MAHA strategy report. We actually have a post dropping on this today. Just really briefly, the report emphasizes shifting children's diets from processed to whole foods. However, the implementation strategy lacks any specificity. So, for example, there's no mention of salt or sodium reduction in this 20-page strategy document, despite sodium being identified as a significant contributor to childhood chronic disease. It also creates new administrative structures, including a new administration for a Healthy America within HHS and multiple NIH offices, while simultaneously reducing funding for existing food assistance programs. On vaccines, the report signals shifts in current recommendations. As we know, Kennedy has already removed COVID vaccine recommendations for healthy children and has brought in researchers to revisit previously studied vaccine safety questions. The report also promotes whole milk over reduced fat options and repeatedly mentions improving access to meat without similar emphasis on fish, poultry, or plant proteins. It also advocates for high quality supplements. I'm using air quotes if you're only listening and not watching video, without defining quality standards. Notable omissions include social determinants of health, healthcare access barriers, climate impacts on food systems, gun violence as a cause of child mortality, and evidence-based addiction treatment approaches. So we will follow up on this. We're doing a deep dive, but it says a lot, but we don't really see any tangible, or specific recommendations for how some of these changes will be implemented. So, okay, now
all right.
let's
Thanks,
get into it. Sarah, do you maybe want to read Dr. Nancy Haigwood's incredible bio for us?
I would, but I actually don't have it in front of me right now. Otherwise,
Oh,
yes.
oh my gosh, that's right. I'm sorry. We had some technical difficulties. Let me introduce Nancy. And just an aside, we are so grateful at Unbiased Science to have Dr. Nancy Haigwood as a contributor. We just put out an article this past week on mRNA because we've received so many questions about this technology. It's been at the forefront of a lot of skepticism because people think that it's this brand new thing when in fact it's been around for a really long time. So let me read a summary of Dr. Haigwood's background. So Dr. Nancy Haigwood holds a BS with honors in zoology from UNC Chapel Hill and a PhD in bacteriology and immunology from the UNC School of Medicine. She completed postdoctoral training in virology at Johns Hopkins School of Medicine with Nobel laureate Daniel Nathans. Dr. Haigwood is a professor in the Division of Pathobiology and Immunology at Oregon Health and Science University with a joint appointment in molecular microbiology and immunology. She previously served as center director and holds a visiting professorship at University of Arizona. Her career spans biotech industry positions before transitioning to academia at Seattle Biomedical Research Institute and the University of Washington. She's a fellow of the American Academy of Microbiology. She has published over 100 peer-reviewed papers. We are so grateful to have you here. Thank you so much, Nancy, for joining us.
Thank you so much, Jess and Sarah. I'm really happy to be here today. I'm looking forward to talking to everyone.
So, Nancy, we're kicking off every episode with a question, which is, what is a news item that caught your attention recently, maybe in the last few weeks or so, and why do you think people should care about it?
Well, I must admit, I've been watching closely the news when it comes to vaccines, and my most concerning news item regards the fact that people will now often need prescriptions in order to get a COVID vaccine. And I think this is a mistake and that needs remedying. I'm not sure how we're going to get there, but that is the thing that's really on my mind. I went out and got my own vaccine because I am over 65. And I was able to get one without a prescription at Costco. And I live in Arizona, so hopefully other people are having better luck. But I have heard a lot of my friends tell me that they've had to go back and get a prescription. And let's not even think. Then there's children.
I'm sorry. I just have a question for either Nancy or Jess. Like, I don't know if either of you know this, but like, does this vary by states? Which state, like, so only certain states require prescriptions for vaccines now?
It's a mess right now.
I can't tell. Do you know? Yeah, it looks messy.
Yeah, no, it's very messy, and certain states, like where I live in Massachusetts, the governor has issued basically a standing prescription for anyone 6 months and older who wants to get a COVID vaccine, but that's not the case in other states. And Nancy, I was going to comment that my mother, who is 70 years old and should qualify for a COVID vaccine based on the recommendations, was turned away from CVS. And it's because there is a lot of confusion right now. And so some pharmacies, including major national chains, are basically putting a pause altogether until this upcoming ACIP meeting, which is scheduled for, actually, I think it'll be right around the time this episode airs. I think it's September 18th, 19th. So we should know more then.
Yeah.
We need to fix it.
Yes.
Sure.
So Sarah, I'm sure, so Sarah nerds out. Nancy, you may or may not know, but Sarah's background is in neurobiology, but she dabbles in immunology and virology. So I don't know, Sarah, do you have any questions to sort of kick off the conversation?
Well, I think since we're going to be kind of picking Nancy's brain about mRNA vaccines, it might be a good idea just to do a quick genetics lesson and biochemistry lesson about like what exactly mRNA is, because it's, three scary letters that maybe we heard in high school biology, but don't actually kind of fully understand. So, most of us have heard of DNA, right? That's our genetic blueprint, the blueprint of life. These are the molecules that make us us. It's a code. And the DNA has to be decoded, right? Because if it's a code, it has to be decoded in order to do what it's supposed to do, to translate what's called the genotype, which is our genes, into the phenotype, which is what we are and what makes us us, our physiology, our makeup. But in order to do that, we have sort of a middleman, and that's RNA or mRNA. The M, I like to say to my students that the M stands for middleman. It doesn't actually, but that like helps them to kind of like remember what it does. It's like it's DNA and then RNA, which is like the middleman, mRNA, and then there's the proteins, and most of us are made up of proteins. Like we're proteins and water for the most part. And so RNA is in every single one of our cells, and it's really important because it's what kind of translates and decodes those genes into our physiology. So I just wanted to kind of like lay that out there just for folks, just to establish like what we're talking about today. And so maybe a good thing now, Nancy, would be for you to explain to us, like, how is RNA incorporated into vaccines? Like we hear mRNA and it's in our bodies, it's in our cells. So how do vaccines use mRNA?
Right. So there's a little bit of interesting history here, because it was a wonderful idea to be able to think about using RNA as a direct vaccine and use the human body as a factory to make the vaccine product, which is a gene from a virus, usually, in this case, SARS-CoV-2. Step back a few decades, and we discovered, and this is a big field of people, although I did work on DNA vaccines, had the idea that we'd like to be able to use either DNA or RNA to get into the cell. Well, DNA is much harder to get into cells because DNA exists in the nucleus and RNA exists in the cytoplasm. So to get DNA from outside into the nucleus, you have to go through two membranes. And that turned out to be extremely inefficient. Even though you could make it work in mice, it didn't work in other mammals or humans. So turning to RNA, fortunately, it was really Katalin Karikó who won the Nobel Prize with Drew Weissman, who together discovered, but really Katalin did a lot of the pioneering work previously as well, that together they figured out how to improve on RNA so that it would not be a signal for bad immune responses in the body. So that was kind of the first step to figure out because she was able to show that you could force RNA into cells, it degrades very quickly, and it caused problems. It caused all kinds of cytokine responses and really destroyed the RNA quickly. So she figured out by making substitutions in the RNA with synthetic chemistry, replacing the uridines with a different form of uridine called pseudouridine, that then this RNA was able to be expressed well, and it worked. But it was still a problem to get it into the cell. And several groups really contributed to this. And I can't remember the name of the scientist who did this beautiful encapsulation work, but basically made a lipid nanoparticle and figured out a couple of different ways to use it, and use 4 different compounds, all of which are non-toxic, to make this bilayer lipid and get the RNA inside, and use that to get the RNA into the cells. And once you've got the RNA into the cells, then it's ready to make the protein of interest, the vaccine candidate. And that really, that's kind of the basics here. There's more to the story that I'd love to touch on, but let me make sure I got to your question first.
I think that was a beautiful answer. My goodness. And I like you say it like so simply, like, oh, the way you're explaining it. And I know there was obviously a lot of decades of research that went into this, but you said you wanted to share something else, or there was something else to the story. So what were you going to say?
Well, the fact that we were able to make mRNA vaccines for SARS-CoV-2, the timing was actually kind of fortunate because not only had Kariko and Weissman been working away on making the RNA better, and this other group had done this fabulous work to do the encapsulation, but the group at the National Institutes of Health in the Vaccine Research Center, led by Barney Graham, was able to make a vaccine for MERS, which is a cousin of SARS-CoV-2. This was never rolled out because it turned out we didn't need to ramp up a MERS vaccine. Fortunately, the epidemic died down by itself pretty much. And so, but it was obvious that the SARS-CoV-2 epidemic was going to be devastating. And I think really they're the ones who started collaborating with Moderna and working with them to make an RNA vaccine for MERS. That stands for Middle Eastern Respiratory Coronavirus, but it's
Syndrome?
Syndrome. That's it. Syndrome.
Oh,
Syndrome.
Yeah. Right.
Syndrome. I beg.
And so it was the cool thing was about Barney was that Barney had been working with another scientist at Dartmouth, Jason McClellan, and he's a structural biologist who figured out how to make the protein, the spike protein for MERS, in a constrained manner. It was so-called pre-fusion because viruses have these proteins that get in, are the key to get into the lock of the cell. And in the process of doing that, they open up. The open up version isn't very immunogenic. The closed version is. So they figured out how to stabilize that closed version for MERS. And so when SARS came along and it turned out it was so close to MERS, it's coronavirus, very similar, they knew exactly what kind of gene to order from the gene synthesis people, which is how we make genes these days. You just order up a gene, but they knew exactly what to order up.
Oh, yeah. Sounds like science fiction. You can just order up a gene, but it's any molecular biologist can tell you that that's just how it works. And they're actually less expensive than you think they would be. So I think because we were talking about how the correlation between the similarity between MERS and SARS. So would you say that that kind of contributed to the speed at which the COVID-19 vaccine was developed? Because we already had all of this kind of background research that we'd done on this very similar virus?
Exactly. And I think, there were kind of two steps there. The first one was the Chinese published the sequence, which was enormously helpful, and everybody jumped on the sequence.
Right.
Fortunately, Barney Graham was paying attention, and his postdoc, Kazmikia Corbett, who actually was part of this study too, she's an amazing person. The two of them together, along with a lot of scientists at the VRC, then used their knowledge that about MERS to make a closely similar vaccine that was SARS-CoV-2 specific.
Yeah. So thank you for catching me up. So there, in the Substack that we wrote, Nancy, we were talking about these other vaccine technologies, right? There are lots of other types of vaccines that protect against other pathogens, diseases. So when people say, why did we use this mRNA vaccine technology for COVID? How would you respond to that?
I would say because it made sense at the time. We knew we had this technology and it had been proven in the lab for other diseases. It just had never been rolled out on a large scale for humans. But with other pathogens like rabies, it had already been tested. We knew it was possible to make a vaccine by this one. Why was that so attractive? Primarily because it's really fast to be able to make an mRNA vaccine compared to every other kind of vaccine, essentially. Any other vaccine, whether it's old-fashioned, attenuated, which you wouldn't want to do with a lethal disease, or a subunit vaccine prepared in the lab, requires scale up, and scale up means, and I did this back in the day when I was working on HIV vaccines, we made a subunit vaccine, but it took us an entire year to make a lot that was large enough to do some clinical trials in phase 1, which is just a very small number of people, because we had to make it, purify it, qualify it, and then show that uh, it would pass regulatory muster. Because we never put anything in people that isn't really tested very thoroughly. All of that against the backdrop of SARS-CoV-2 epidemic, we could see from looking at how the virus was changing over time that we were going to have a different virus a few months later. And that turned out to be true. And as you know, people monitored the types of cases that came into the hospital. They also monitored waste streams to look at the changes in the genetic sequence of the virus. And those changes affected the spike protein in the vaccine. So that's the most variable part. That's how the virus gets away from the immune system, by mutating its spike protein and a few other things. But primarily, that's where all the changes are, because it can evade. And it's an RNA vaccine. That's what RNA viruses do. So we knew what we were up against. We knew we had a technology that could make a vaccine very quickly. And it was really, I would say, the only choice to get a vaccine in short order, meaning within a year. And fantastically, it was able to be made, manufactured, tested in animals, and then moved on into clinical testing, safety testing. And because we had an epidemic, it was possible to tell the difference between the vaccinated people and the ones that got the vaccine and the ones that got placebo. So I kind of jumped ahead, but basically the RNA was really technologically good, and also immunologically. It was a great choice because it's made in the body, it can make all of the immune responses that would normally be made just as if you had an infection. So you're going to make the T cell responses and the antibodies. And both of those can be very important.
Nancy, you addressed something that I was going to ask you about, which is, there's a lot of skepticism around how are we able to create this so quickly? And for all the reasons that you've outlined, and the fact that there were all eyes, right? It was a global pandemic. So everyone was working together. There was obviously a lot of funding that allowed us to carry out the necessary phases of research. And as we wrote in the Substack, no steps were skipped. And I think this is a really common misconception, right? Steps were not skipped. Many of them happened in parallel with overlap, instead of happening consecutively, because there was this unbelievable urgency, right? So I think that was a really important point to make. Another thing that we hear all the time when we're talking about mRNA is, well, isn't this messing with our DNA? Can't this somehow alter our DNA and be harmful? And we've seen people say things like, oh, it can cause cancer and other negative outcomes. So how would you respond to that?
It's an important question, of course. But RNA doesn't do those things. The only type of RNA that causes a problem is the RNA of a retrovirus that goes the other way. And HIV happens to be an example of a retrovirus that comes in as RNA and brings with it its own enzyme to turn that RNA into DNA. This does not happen except in these limited cases of retroviruses. Retroviruses are deadly, deadly viruses. And once they're in your DNA, you've got them forever. Almost all the rest of the virus families make what's called an acute infection, and then they're gone. That's because they aren't messing with our DNA. RNA does not make DNA, and it gets degraded pretty quickly, usually within a day, I would say, for the most part. I don't know the precise numbers, but it's a short-lived molecule, and that's how it's designed to be. And really, this makes RNA vaccines much safer, in my view.
Yeah, absolutely. I mean, it's kind of like what I was saying at the beginning of the episode about how like mRNA is kind of that middleman. So it doesn't make DNA; it actually comes from DNA. So unless you have this, like you were saying, Nancy, this retrovirus that actually retro meaning it goes backwards, so it takes that RNA and then transcribes it back and then goes backwards back into DNA. most mRNA of most viruses, like you were saying, they don't do that. So they only go in one direction. Like most often, it only goes from DNA to RNA. It does not go from RNA back to DNA. So that's an important thing to keep in mind.
Right. Humans do not have an enzyme to do that. So we can't do that. We're not going to do that.
So, Nancy, I'm sure you were horrified. I was horrified with the news of the $500 million of funding cuts to mRNA research, although it seems to be specific to mRNA vaccines for respiratory illnesses, COVID, avian flu. And that in and of itself is very problematic, of course, because we need to be prepared, against these illnesses. But can you talk about the incredible potential of mRNA technology against other things, other diseases like cancer?
Well, I'm not a cancer biologist, so I'm a little hesitant to move into that field, but I can definitely see that having the ability to get a small piece of DNA from one source and make a product in a human body, whether it's an anti-cancer molecule or a vaccine, is incredibly powerful. And we as a field of scientists and doctors have just been so fortunate that all this work for the last 40 years and last 100 or so years has led to this point where we have the flexibility, the manufacturing capability, and now we have tremendous safety data from SARS-CoV-2 vaccines to where, especially in cases where there's a lot of variation, like influenza or SARS-CoV-2 or many other viruses that are highly variable, or in an epidemic situation when you need a vaccine really fast, you'd like to be able to use this mRNA approach. And I'm really puzzled, frankly, as a scientist, why the consternation? Where is this coming from? Because the data are really very strong that this has been used in literally hundreds of millions of people now for SARS-CoV-2, and the safety data are very, very strong. So it saves so many lives to do this. Why would we prevent ourselves from taking advantage of all of this technological advance? I just don't get it.
Well, I don't either. And really, unfortunately, I think a lot of it stems from people really not understanding what this technology is and the power of it. And that's why I'm so grateful. We're so grateful that you're helping us communicate that to the public. So we're starting to get to the end of the episode here, but I think we'd be remiss if we didn't ask you to just tell us a little bit. Just before we hit record, Nancy just casually dropped that she was part of a team that was working on an HIV vaccine. Can you just tell us a little bit about your work?
I did that quite a long time ago. It was 40 years ago, but I'm very pleased to have been part of a group that developed this subunit vaccine for HIV called GP120. And we had competition. There were other groups doing similar work at Genentech. I was at Chiron at the time. And sadly, that particular subunit, although it produced antibodies, it couldn't produce T cells. And we didn't see the kinds of immune responses that we were hoping to get. In hindsight, these stabilized proteins that I was describing to you that have now been made for respiratory syncytial virus and for MERS and SARS, these are examples of stabilized proteins that appear to be much more immunogenic because they're in that closed conformation, and that's really where the virus is most vulnerable. We think that's probably the case for HIV as well. And even though it's been 40 years of research, a lot of the research that led to these discoveries of these new vaccines was funded by HIV research, which we learned a lot about adjuvants. We learned about viral vectors. We learned about lots of different ways to make vaccines that didn't work for HIV, but they gave us clues of how they could work for other viruses. And so I'm hopeful that some of the newer ideas that are being tested right now will come to fruition. There's some promising studies. Personally, I just want to say a couple of things. I got excited about antibodies and what they could do, and there's a whole other field of therapeutic antibodies for infectious diseases, and some fantastic work by a number of wonderful scientists, many of whom would probably love to come on talk to you on this show, so I'll recommend them. But we have taken advantage of their fantastic work to discover antibodies. We now have this treasure chest of monoclonal antibodies from humans infected with different diseases, and we can pick and choose exactly which ones we want to test. And we have shown that when you give these antibodies to baby monkeys infected with HIV cousin, that even after the baby's infected, you can come in with these antibodies and treat them within a few days of infection, and the infection will be cleared. And the animal does not need any drugs or any treatment. So it's an alternative method for curing HIV, but you got to get there early. So again, this is another story. I'm happy to come back and talk about that. But I also think you might want to talk to other scientists who are in the antibody discovery field, which is really where it's at. It's a combination of intense study of pathogenesis and unbelievably beautiful structural work that's been done to understand how proteins fold. It's an exciting field, and
I got to tell you, Nancy, I'm getting, everything makes me cry. If you know me, I cry at the drop of the hat, but I get very emotional hearing, honestly, I am just blown away by science, and how far we've come. And I think it's incredible. And hearing someone like you just share, first of all, share your time and expertise with us, but just the way you talk about it and the incredible, we talk about these things, but I want people to remember we're talking about diseases that have killed millions of people. Like these are incredible technologies and advances that will save countless lives. It absolutely takes my breath away. I can't thank you enough. Nancy, we like to end every episode on an optimistic note, and we're wondering, is there something that's giving you hope with regard to health and science right now that you can share with the audience?
Oh gosh, there's so many things that are going well. You mentioned cancer. Look at survivability now, and the fact that we can modulate the immune response in ways that can go after cancer cells. I'm a cancer survivor, so I'm a huge beneficiary of some of this technology. There's so much exciting going on in medicine today, and I've been fortunate to be able to work in basic discovery research for so long, and I can't quit. So that's why I'm here, and that's why I love talking about science. And I do think we need to emphasize these positive breakthroughs. There are so many. So thank you again for inviting me to come today. It was my fun. I had a lot of fun.
Yeah.
Here come the waterworks. No.
Yes. Classic. Classic Jess.
Thank you. Classic Jess. No, really, Nancy, thank you so much. Thanks for contributing to Unbiased Science. And to our audience, you can follow along. Nancy will be bugging her to continue to contribute. And I really am hopeful that our technology issues don't interfere with the message here today and the incredible information that Nancy shared. Sarah, would you like to take us home?
Sure would. Thanks for tuning in, everyone. We hope you learned a thing or two. Please join us next week on your trusted source for no nonsense, just science.